this presentation and the Giulio Gabbiani lecture. The topic is a vast topic, cardiac fibrosis. So I will clarify a little bit what I will be discussing today. So I will provide first some definitions. Then I will discuss the cell biology of cardiac fibrosis, focusing on fibroblasts, myofibroblasts, but in particular pericytes, which has been a cell of major interest for the lab over the last few years. I will discuss the functions of fibroblasts in myocardial disease and their diversity of functions that they exhibit. And I will close with some therapeutic perspectives. So we do not usually pay attention to the cardiac interstitium, but unless there is disease. And this is because practically in every type of myocardial disease, there is expansion of the cardiac interstitium through deposition of extracellular matrix proteins. And this is what we call cardiac fibrosis. And this is a response. It's not a distinct disease, in contrast to the lung, where there is idiopathic pulmonary fibrosis, a primary fibrotic condition, in contrast to scleroderma, which involves the skin and other organs, which is a primary fibrotic condition. There is no described primary fibrotic condition involving the heart, but it is a response that accompanies all forms of myocardial injury. And it can be reparative or it can be maladaptive. And with simple histology, one can obtain useful information to distinguish between the two. So when you have replacement fibrosis, where a large number of cardiomyocytes die, there is a formation of scar, which is almost always replacement fibrosis, reparative in nature, fills the hole left by the cardiomyocytes in a non-regenerative organ, and the mammalian heart in adult mammals is a non-regenerative organ. On the other hand, in many other conditions, including conditions associated with pressure loads, such as hypertension, valvular diseases, metabolic dysregulation, there is accumulation of matrix in the interstitium, and this is interstitial fibrosis. There's also perivascular fibrosis, and these can be maladaptive, and they can exacerbate dysfunction. And this is why we're particularly interested in cardiac fibrosis, because there is evidence that fibrosis can cause dysfunction, and also can be involved in the pathogenesis of conduction defects and arrhythmias. The evidence in human patients is associative. So in many different myocardial conditions, the severity of cardiac fibrosis associated with mortality and adverse outcome. But if we use the insights gained through animal models, there is sufficient evidence to support that fibrotic remodeling can cause or exacerbate dysfunction, both systolic and diastolic. Systolic mostly when it's associated with matrix degrading processes, which result in loss of signals that support the cardiomyocytes, their survival and function, and may lead to progressive cardiomyocyte death, dilation, and systolic dysfunction. And the diastolic dysfunction because of the fact that the heart becomes stiffer, less compliant when there is deposition of cross-linked collagen. And the importance of cardiac fibrosis in conduction, in the pathogenesis of conduction defects and arrhythmias is an area of active research, as AJ alluded to. A recent study by Arjun Deb in Science suggests that actually fibroblasts are not involved in the pathogenesis of arrhythmogenesis just by depositing collagen and just by causing blocks and facilitating re-entry circuits, but also they can couple with cardiomyocytes, and they can trigger excitation and arrhythmia generation and propagation. So these are the cellular effectors of fibrosis, practically every cell that you can find in the heart. There is a big difference, though, because all these cells in the top row, immune cells, vascular cells, cardiomyocytes, platelets, they can contribute by producing cytokines and growth factors, matricellular proteins. They can produce protease that remodel the matrix. What they do not do is they do not produce structural matrix, which is the most important component of fibrous tissue. Only fibroblasts and their activated form myofibroblasts are capable of producing large amounts of extracellular matrix proteins. That's why these cells are the key effectors, are considered the key effectors of fibrosis and of cardiac fibrosis in this discussion. So it is important to define what a fibroblast is. And many years after their first description, fibroblasts remain poorly defined. So we still use morphological and functional criteria to define that fibroblasts are cells of mesenchymal origin that reside in connective tissues, lack a basement membrane, and secrete extracellular matrix proteins. And this, unfortunately, has not changed much since the early description of the fibroblast, which dates back to the mid-1850s when Rudolf Virchow described them. So this is a page from the book where they are described. He didn't use the term fibroblast, but he used the term Spindelzelle des Bindegewebes, which means spindle-shaped cells of the connective tissue. And this is a drawing, because this is how information was communicated in this time in pathology, using drawing of fibroblasts in tissue. He described them as cells producing extracellular matrix proteins. And unfortunately, there is not a single specific and reliable marker of fibroblasts. So their identification is typically based on a combination of positive markers and exclusion criteria. I've included here a table from one of our reviews that attempts to describe the positives and the negatives of each one of the markers that have been used to identify cardiac fibroblasts. I didn't include it here to actually go over it, but just to illustrate that whenever you need to provide so much information to give an idea on which markers should and should not be used, there's probably not an optimal marker. And this is why I like using this slide from Sylvia Evans' work from Moore Morris and the JCA, which does exactly that in tissue. So you can see here they're using a collagen reporter mouse to identify the fibroblasts. And in addition, they are using staining for PDGF receptor alpha, which is a relatively specific marker for fibroblasts, vimentin, which is a general marker for mesenchymal cells, and then some exclusion markers. So fibroblasts are non-endothelial cells, so PKM1 negative. They are non-hematopoietic cells, they're CD45 negative. They may express PDGF receptor beta, which is a mural cell marker, but is expressed by about 50% of cardiac fibroblasts in our experience. And they express alpha-massalactin only when they're myofibroblasts. And a simplified form of this that you will see throughout the talk in our work, we're using the PDGF receptor alpha reporter mouse, which is relatively specific for fibroblasts, and we're using, in addition to it, alpha-massalactin staining to identify myofibroblasts as PDGF receptor alpha positive, alpha-massalactin positive cells. And you can see how these expand following infarction and how you can appreciate the very large number of fibroblasts. And this is a nuclear reporter. That's why the fibroblasts have this appearance. And the myofibroblasts, which are a fraction of the fibroblasts in the infarcted heart. I mentioned the term myofibroblast, and the question is, what is a myofibroblast? This was a seminal discovery in the cell biology of fibroblasts made by Giulio Gabbiani and his colleagues about 50 years ago. So Dr. Gabbiani identified the myofibroblasts. First, they published their work in a paper in Experiencia. That was the first description. And then this was followed by a paper in Science, which uses for the first time the term myofibroblast. It's a modified fibroblast that expresses cytoskeletal proteins, which are typically found in vascular smooth muscle cells. These are contractile proteins. You can see here, this is one of the very nice pictures that Boris Hintz produces, the incorporation of alpha-massalactin into the stress fibers. It's the red staining in this large myofibroblast. And these cells are typically found in wounds. They are contractile cells, so they're involved in contraction of scars, but they also have high activity in producing extracellular matrix by association. And for this reason, they're important in fibrosis in many organs, including the heart. There are two major characteristics of fibroblasts that have been appreciated, which are important to understand their biology. The first is their heterogeneity. It's not a single cell type, but there are several different subpopulations of fibroblasts, and single cell studies have contributed to realizing and understanding these classification of fibroblasts into various subsets. This is from Alex Pinto's work from Australia, and you can see the various clusters here. It's important to emphasize that in contrast to other cell types, such as lymphocytes, which are polarized cell types, which you have polarized subtypes that you can clearly identify, and both from a transcriptomic and functional perspective. In fibroblasts, there is a palette of nuanced colors. So there is a continuity in these phenotypes, and it has been difficult and challenging to associate specific subpopulations of fibroblasts from transcriptomic studies with specific functions. So some of these are quite consistent between studies. If you study the same organs, or for example, the fibroblast SILP, the high SILP expression or the high THPS4 expression. However, their functions have not been identified. So the field lacks this connection between transcriptomic profile and functional phenotype, which would be very important. And the second characteristic that fibroblasts have is their phenotypic plasticity. So they change their morphology, their function rapidly, depending to their environment. And anyone who has cultured fibroblasts will tell you that, but this is from our own experience and our own work. So if you culture them in culture plates, they become myofibroblasts. You can see here the alpha-smacalactin decorating the stress fiber in this large myofibroblast. Then they resemble the cells that you have in wounds and in fibrotic tissues. If you take these cells and you put them in a collagen lattice, in a collagen pad, in this environment, the collagenous environment of the pad, the cells in free-floating pads in this case, the fibroblasts lose their alpha-smacalactin expression, they disassemble their stress fibers, and they become more like these dendritic-like cells with very little expression of alpha-smacalactin. They produce a lot of MMPs and TIMPs, and morphologically, at least, they resemble a lot the fibroblasts that you have in normal tissues. And between these two extremes, there are various subtypes that you can have depending on the environment in which the fibroblasts are subjected. There is no better model to understand these transitions of the fibroblasts and the changes in their phenotype than myocardial infarction, because in myocardial infarction you have all these dynamic changes in the environment that drive also changes in the fibroblast profile. So early on, the necrosis of a large number of cardiomyocytes induces inflammation, and the fibroblasts behave at this stage as inflammatory cells, and they produce also MMPs and they degrade the matrix. Then during the proliferative phase, that's when you have myofibroblast conversion, and that's where you have proliferation of the fibroblasts and expansion of their numbers. And finally, the more enigmatic of the three phases, the maturation phase, during which the fibroblasts lose their myofibroblast characteristics. They may become apoptotic, but the majority of them remain there in the scar, and they acquire some matrix preserving properties, expressing genes associated with tendons, as has been described by Jeff Molkent in Healing Scars. This is from our early work on the presence of myofibroblasts in the infarcted heart, and you can see that this is in a canine model, the abundance of myofibroblasts identified as alpha-smooth muscle actin positive cells, organized in nice well-aligned arrays of cells in the border zone, which are myofibroblasts and not smooth muscle cells, because they express alpha-smacalactin and the embryonal isoform of smooth muscle myosin, but not mature smooth muscle myosin proteins, such as isoforms of smooth muscle myosin, SM1 and SM2. You can see that the numbers increase 5 to 14 days after infarction, a reperfused infarction model. So they peak at that stage, and then they're reduced afterwards. So there is a marked expansion of myofibroblasts in the infarcted heart. And there is also a marked expansion in fibroblasts in general in the infarcted heart, several-fold increase in their numbers. So a question that comes up is, where do all these cells come from? And this is back when I was a fellow in Mark's lab, when I was actually very unsophisticated. I was a clinical trainee, and I joined the Mark's lab to do the clinical investigator pathway, knowing very little about cell biology, but then things were much simpler for me at that time. And I thought that the answer to this question is very simple, actually. So we had the fluorescent microscope with a camera hooked up to it, and I could take nice pictures, and I could do the pathology to the level that I could, and I could see fibroblasts in the heart. I could see a lot of fibroblasts and myofibroblasts in the infarct, and there was evidence that these cells would proliferate. So this is staining for the proliferating cell nuclear antigen, and this is alpha-muscle-actin staining. So you have a lot of proliferating myofibroblasts. Not a surprise, because there are a lot of proliferative growth factors in the infarcted heart. So that's where the fibroblasts come from, and all the myofibroblasts in the infarct. So you have resident cardiac fibroblasts differentiating into myofibroblasts and proliferating due to the effects of the abundance of growth factors that you have in the environment. So a very straightforward answer. However, over the next 20 years, there was a lot of evidence suggesting exotic sources, additional sources for fibroblasts and myofibroblasts in the infarcted heart, and in general in fibrotic tissues. So there was suggestions that immune cells, in particular bone marrow-derived cells, myeloid cells in some cases, some people went further to suggest that there were macrophages that they actually could convert into fibroblasts. There was evidence on the endothelial to mesenchymal transition, which was documented in several different organs and in the heart as well. So another contribution from endothelial cells. So all this became a rather controversial area, as with the advantages that came with the use of lineage tracing approaches. There were conflicting results between various studies, depending the relative role of these other populations. So what is the basis for the conflicting data? There are two major reasons, and one is the use of non-specific Cre drivers to trace the different cell types. That's particularly important in endothelial cells, where the early type 2 Cre drivers are not specific for endothelial cells, but also for some of the other cell types. The second one, and the major one, is the criteria used for identification of fibroblasts. As you saw early on, there is not a single marker that you can use to identify a fibroblast. So there will be some subjectivity in suggesting that there is a transition of your cell to a fibroblast, and studies that use non-specific markers tend to support conclusions on cellular plasticity and fibroblast conversion, whereas studies which are more strict and more restrictive on the definition of fibroblast identity may not. So an example here on the question whether myeloid cells and hematopoietic cells in general can undergo fibroblast transdifferentiation, three different studies. The first one by Sylvia Evans using a VAV-Cre line to trace hematopoietic cells, and a collagen reporter mouse to label fibroblasts, suggests no. The second one using a lysozyme M-Cre to trace myeloid cells, and a periostin transgene to identify the myofibroblasts, and also a number of immunohistochemical labels to label the fibroblasts. There's a study by Jeff Molkentin's group, by Onur Kanisichak, also says no. Then there is a study in the JCA by Jagat Narula, which suggests using the same exact Cre driver, the lysozyme M Cre driver, that they can convert. However, the criterion for identification of fibroblasts is immunohistochemical, and uses some markers which may or may not be very specific. Plus, also it is prone to the artifacts that you can have with immunohistochemistry. So FSP1, fibroblast-specific protein 1, is used in this study, and it is a marker that does not just label fibroblasts, but also several other cell types, including macrophages. This is our contribution to the field of macrophage to fibroblast conversion, and this is work that we have not yet published. We actually did it because we wanted an answer to this controversial topic, and we used a reporter mouse for define fibroblast identity, and we used the CX3CR1 CRE driver to identify macrophages, and we found virtually no macrophage to fibroblast conversion, and there was a very, very small number of cells in control and infarcted hearts that were derived by CX3CR1 positive cells, and that suggested that there was no significant conversion of macrophages to fibroblasts in the infarct. Importantly, nowadays, I believe that we need to expand a little bit our view of cellular identity, because using a single marker is very restrictive. And the single cell approaches give us a great tool in understanding changes in identity and in cell state by studying a range of genes, because one could argue that there may be conversion of fibroblasts which may not be defined just as a PDGF receptor alpha positive cell, but through expression of other genes. And again, if you do single cell analysis, here we did the single cell analysis of the CSF1 positive cells, which are myeloid cells in the infarct, you can see that they express large amounts of vimentin, but they do not express any fibroblast markers. So they don't express TCF21, they don't express FAP, they don't express PDGF receptor alpha, which are the more specific of the fibroblast markers. Importantly, they don't express much collagens, at least not structural collagens. They do express a lot of S100A4. Why do I emphasize S100A4? Because it's FSP1, that fibroblast-specific protein 1, which many people still use as a marker of fibroblasts, despite the fact that it is well demonstrated that it is expressed in other cell types, including macrophages. So macrophages may not convert into fibroblasts if one uses strict criteria for cellular identity, and they don't contribute to collagen expression, but our single cell studies suggest that they contribute to fibrosis by expressing growth factors and also a range of matricellular proteins. So amylin-2, thrombospondin-1, SPARC, osteopontin, ECM1, versican, fibronectin are all highly expressed in macrophages in comparison to other cell types. And through the production of these mediators, these matricellular proteins, the macrophages can change the environment and make the fibroblasts more fibrogenic. So most activated fibroblasts and myofibroblasts in infarct and remodeling hearts are derived from resident fibroblasts and not from blood-derived or from endothelial cells. As you notice, this statement leaves out a population of cells that has remained enigmatic for many years. These are the pericytes, and we're particularly interested in the pericytes. And here, these were two of the major stimuli that increased our interest. There was a study by Raphael Kraman from Benjamin Humphrey's group showing that the perivascular GLI1 positive progenitors are key contributors to organ fibrosis, several different organs. And these cells are sometimes called pericytes, or it's unclear what their identity is. But they gave, based on that study, a large proportion of the activated fibroblasts in various organs. And then if you look at the atlas of the heart by Teichmann, you can see the abundance of neural cells in human hearts. So these are pericytes and smooth muscle cells, and they appear to be even more abundant than fibroblasts, at least with this algorithm and protocol that was used here. So we were particularly interested in pericytes, and Linda Alex worked on this project in the lab. And first we needed to characterize our tools. And after looking at several different markers and CRE drivers for the pericytes that would distinguish them from fibroblasts, we identified the combination of NG2 as a pericyte marker, as a neural cell marker, and PDGF receptor alpha as a fibroblast marker, because this identifies neural cells and fibroblasts as two separate populations. histologically and also flow cytometrically. And when you look at the transcriptome is consistent with what you have one would have expected for fibroblasts and the pericytes. So we moved on to use inducible NG2 Cre drivers to study the fate of pericytes in various models. You can see here the strategy, the breeding strategy, and the fact that we used a reporter line, the PDGF receptor alpha reporter line, to reliably identify fibroblasts histologically, because we did not trust existing antibodies for fibroblasts. This is a protocol that includes the DB phenotype, because we have used this in a diabetic model as well. But this same approach we used in the other models of myocardial disease, including infarction, which is the data that I will show here. So this is the lineage tracing of the NG2 lineage cells, of the mural cells. These are labeled as red, and the green is the PDGF receptor alpha positive fibroblasts. These are nuclei. It's important to emphasize this to interpret these images. And you can see that the majority of these PDGF receptor alpha positive cells are not derived by mural cells. However, there was a small percentage, about 1 in 20, so about 4% to 5% of the fibroblasts in the infarcted heart that were also derived, that were also NG2 positives, that were derived by NG2 positive cells. So it seems that there is a contribution, at least in the infarct, of the NG2 lineage cells to the infarct fibroblast population. A small contribution, but it is there. And this is not something that we saw in the other models of heart disease. So we did not see that in the diabetic mice, and we did not see that, and we haven't published that last part in the pressure overload model. So it has to be something, some stimulus that is specifically activated in the presence of extensive necrosis with an intense inflammatory response that you have following infarction. And to examine this further, and because, as I mentioned, nowadays single cell work is important to understand changes in identity, we sorted these NG2 lineage cells when we did the single cell RNA-seq on these NG2 lineage cells. And we had the following observations. First of all, the NG2 lineage cells expanded after infarction. You can see this is the graph that shows this, much darker levels in the infarct versus the control. And also the cells diversified in the infarct. So the brown color here shows the various clusters that were identified in the infarcted heart, whereas the light blue shows the clusters identified in the control hearts. And you can see that there is appearance of new clusters following infarction, which is independent actually of the increase in the numbers that you have. And actually, one of these clusters, cluster 7, which was a sizable cluster post-MI, was about 20% of the NG2 lineage cells, exhibited expression of fibroblast identity genes, several of them. So TCF21 was high, PDGF receptor alpha was high. So these appear to have acquired, these cluster 7 cells appear to have acquired a fibroblast-like phenotype. And we used also pseudotime trajectory analysis to infer transitions between the cells in the infarct. So this, depending on the transcriptional state of each cell, in a computational, with computational strategies, it infers how the cells may have transitioned. And they seem to have been derived from a population of cells that were in cluster 8. And these cells, the characteristic that they had is that they were proliferative cells. They had high expression of proliferation genes. Thus, it seems that the paradigm that is supported by the histological studies, lineage tracing studies, and the single cell studies, there is a low-level contribution of pericytes to the fibroblasts, that about 4 to 5% of the fibroblasts are derived from the pericytes. And this is driven by proliferation of the NG2 lineage cells, of the mural cells, of the pericytes in the infarcted heart, that also contributes this population of activated fibroblasts. We then asked whether there is some unique feature functionally of these pericyte-derived fibroblasts, and we sorted them from infarcted hearts. So these graphs show matrix gene expression, and the middle bar, the PF, is the pericyte-derived fibroblasts, whereas the P here is the pericytes, and then non-PF is the non-pericyte-derived fibroblasts. And as you can see, these pericyte-derived fibroblasts seem to be overactive, even in comparison to all the other fibroblasts. So they express more matrix genes, more MMP2, more collagen-1, alpha-2, more TIMP2. Several of the profibrotic genes are higher in these cells. So they seem to be overactive. So perhaps their contribution may exceed what is suggested, the limited impact that would be suggested by their low numbers and by the low contribution to the fibroblast expansion. And important contribution that neural cells make following infarction is in angiogenesis, and specifically in vascular maturation. So you can see here, this is lineage tracing in the infarct. You can see in red these NG2 lineage cells as they become incorporated into the neural wall and as they interact with existing neural cells, and they form this thick walled vessels in the mature scar, which may not serve as conduits of blood because they are in a scar that has a lot of collagen around it, but may serve to stabilize the scar, preventing progressive infiltration of the infarcted heart with leukocytes, preventing and control angiogenesis. So an important contribution of the pericytes in the infarcted heart is to form these mature vessels, so to coat these forming neovessels after myocardial infarction. Then we asked how are pericytes activated, and our focus was primarily on the fibrogenic effects of the pericytes, and because in vitro we had data that at least in human pericytes, TGF-beta induced a fibrogenic profile. It induced expression of several fibrosis-associated genes. We generated mice with pericyte-specific loss of TGF-beta signaling. So these are mice that lack the type 2 TGF-beta receptor 2, specifically in pericytes. It's the receptor that transduces signals from all three TGF-beta isoforms. So you have no TGF-beta signaling. And what we found, somewhat surprisingly, is that there was no impact of pericyte-specific TGF-beta signaling loss on the fibrotic response and on the reparative response, on the myofibroblast response following infarction, but there were effects in vascular maturation. So these animals had perturbed vascular maturation, which resulted in worse remodeling. So it seems that pericyte-specific TGF-beta signaling is protective by driving vascular maturation. So the conclusions from this study, which was published last month in Circulation, is that in the infarcted heart, a subset of pericytes converts to fibroblasts and accounts for 4 to 5% of infarct fibroblasts, that pericyte-derived fibroblasts are overactive, expressing large amounts of extracellular matrix genes, that pericytes contribute to infarct angiogenesis by coating infarct neovessels, and that the vast majority of infarct fibroblasts and myofibroblasts are derived from resident fibroblasts. Okay, so they're derived from resident fibroblasts. However, what do they do? What is the function? What is the role of fibroblasts in myocardial infarction and in heart failure in general? And to understand this, one has to look at fibroblasts in each one of the phases of cardiac repair, starting first from the inflammatory phase. And regarding the inflammatory phase, there is a lot of in vitro evidence and associative evidence suggesting that fibroblasts can behave as inflammatory cells. They activate the inflammasome. They can produce cytokines, produce a lot of CCL2, MCP1, they can produce cytokines. However, many other cells can do the same thing in the infarction. Endothelial cells produce a lot of chemokines and cytokines. There are macrophages, other macrophages that will influx monocytes that will become macrophages that infiltrate the infarct. There are neutrophils, there are cardiomyocytes. So the relative role of fibroblasts as inflammatory cells in the infarcted heart is very poorly understood because the studies that look at cell-specific knockdown of key pathways involved in inflammation have not been performed. There is a lot of work on the role of fibroblasts as reparative cells in infarction. I will show the work that came from our lab, focusing mostly on the TGF-beta system and on the TGF-beta signaling cascade. As you know, TGF-beta is a growth factor that is present in significant amounts in normal tissues, including the heart, in a latent form. It's activated after infarction. There is a release of active TGF-beta. There is de novo production of TGF-beta. TGF-beta binds to its receptors and then signals through SMAD-dependent and SMAD-independent pathways. And our work with knocking down some of the key TGF-beta downstream of TGF-beta pathways, such as SMAD3, specifically in myofibroblasts, supported the notion that fibroblasts and myofibroblasts are protective in the infarcted heart. Here, the myofibroblast-specific SMAD3 knockouts exhibited late increase in rupture. And you can see the rupture tracks here as we section the heart from base to apex to identify the channel, the rupture channel. And this is increased when the myofibroblasts lack SMAD3. And this is because with these disrupted myofibroblasts have alterations in their shape and function, so they're more round, they're malaligned, so the tractional forces will be reduced, and this will predispose to adverse remodeling and dilation in addition to the effects on the rupture. And also you have changes in the morphology of the extracellular matrix with disrupted collagen fibers that contribute to the reparative defect. And there are further defects due to the perturbations in reparative fibroblasts. So this is work that Isabella Tuletta did recently in the lab. So she knocked down a molecule upstream of SMAD3, which actually is responsible for the transduction of signals from all three TGF-beta isoforms, TGF-beta receptor 2. So she knocked it down specifically in fibroblasts using the collagen 1 alpha 2 Cre driver, the inducible collagen 1 alpha 2 Cre driver. And these animals predictably also had cardiac rupture, but also there was a surprising finding during the maturation phase in the animals in the fibroblast-specific TGF-beta 2 knockout mice that survived through infarction and through the reparative phase. There was deposition of fat in the heart in the form of adipocytes. So there was, you can see here in the controls, there is formation at 28 days of a scar that contains mostly cross-linked collagen, whereas in the fibroblast-specific TGF-beta 2 knockouts, there were large pockets of adipose tissue. These are staining with perilipin demonstrating this. They are likely derived from conversion of fibroblasts to adipocytes. Thus, it seems that TGF-beta signaling is important to maintain the fibroblast phenotype as a matrix-preserving cell and to prevent conversion into other cell types in the environment of the infarct, which contains growth factors and cytokines that can guide these mesenchymal origin cells into many different directions. So in the infarcted heart, activate fibroblasts and myofibroblasts play a central protective role, and repair, that's not perhaps too much of a surprise. The non-regenerative organ, you have the hole that is created by losing cardiomyocytes. There's not much of regeneration. You need something to fill that hole. However, one would argue that in conditions which are not associated with extensive cardiomyocyte loss, the activated fibroblast may have mostly maladaptive functions. That was not exactly our experience. So we identified protective effects of fibroblasts even in models of pressure overload, such as the one shown here. This is the transverse aortic constriction model, in which as we constrict the transverse aorta, the heart is subjected to a pressure load and exhibits early on hypertrophy, fibrosis, and diastolic dysfunction. And then late, weeks weeks to months, many weeks to months after the transverse aortic constriction, depending on the gradient achieved. There is dilation, matrix degradation, and systolic dysfunction. So when we looked at the effects of fibroblast-specific SMAD3 loss in these animals, we identified the protective action of the fibroblasts and specifically SMAD3 signaling in fibroblasts in this model as well, which was an early effect related to matrix preserving actions of the fibroblasts. So early on, TGF-beta activated fibroblasts, they suppress the levels of matrix metalloproteinases. They produce inhibitors of metalloproteinases, thus preventing generation of matrix fragments, which can be pro-inflammatory, and also by preserving the matrix network around the cardiomyocytes, which is important to prevent annex, death of cardiomyocytes. And thus, in the early stages of pressure overload, the fibroblasts and myofibroblasts play an important protective role. So in the infarcted heart, there is a protective effect of myofibroblasts. In the pressure overloaded heart, there is also a protective effect of the myofibroblasts. So if they have these reparative and protective actions, why do they have a bad reputation as the cells responsible for cardiac dysfunction? And why does everybody would like to inhibit them and actually delete them from the injured heart? Well, this is because unrestrained, excessive, or inappropriate activation of fibroblasts, chronic activation of fibroblasts, results in progressive fibrosis and may promote ventricular dysfunction, may exacerbate ventricular dysfunction, both systolic and diastolic. And an example here from our own work, this is work that recently Claudio Humeres did in the lab, is Claudio looked at the role of the inhibitory SMAD7 in fibroblasts and myofibroblasts. So this is a protein that inhibits TGF-beta signaling cascades. It serves as a negative feedback mechanism to restrain TGF-beta signaling. And thus, Claudio generated mice that have myofibroblaст-specific loss of SMAD7. And these animals have increased mortality, not because of rupture, but actually because of heart failure. And they develop progressive and extensive fibrosis in the border zone involving the papillary muscles. And then Claudio also went on to examine the mechanisms of SMAD7 actions. He identified the level at which SMAD7 interacts with the TGF-beta cascade, which is downstream of the TGF-beta receptors, because as you can see here, when you have SMAD7 null cells, stimulated with TGF-beta, their levels of activation of TGF-beta receptor 2 and the TGF-beta receptor 1 are comparable with the control animals. However, levels of phosphorylated SMAD2 and phosphorylated SMAD3, but not total SMAD2 and SMAD3, and also levels of certain of the SMAD-independent cascades, levels of activation of some of the SMAD-independent cascades are increased in the absence of SMAD7, suggesting that the level of interaction is downstream of the receptors. And then, and that was the major contribution of the study that was published last year in the JCA. Claudio identified the novel TGF-beta independent mechanism for SMAD7 actions. We identified this with transcriptomic work first. So we did RNA-seq, and this suggested that receptor tyrosine kinase pathways may be overactive in the absence of SMAD7. Then he did a proteomic array, and he identified specific RTKs which may be overactive, and in particular he focused on ERBB2 which is overactive in the absence of SMAD7, and he documented an interaction between SMAD7 and ERBB2. And then he found that part of the effects of SMAD7 on cardiac fibroblasts are not due to actions on the TGF-beta system, but actually on this inhibitory action on ERBB2 signaling. So to return to the role of fibroblasts, fibroblasts in the remodeling heart have protective functions, but need to be tightly regulated. The summary of all this work is that fibroblasts have a wide range of functions in diseased hearts. They can activate inflammatory pathways, they can deposit matrix, they can regulate matrix remodeling, they can protect cardiomyocytes. Other people's work has shown that they may promote angiogenesis as well. They may couple with cardiomyocytes, electrically couple with cardiomyocytes and participate in excitation and erythmogenesis. Following myocardial injury, fibroblast activation is important in repair. However, restrained, prolonged, excessive fibroblast activation contributes to the pathogenesis of heart failure. Important questions in the field relate to the impact that the heterogeneity of fibroblasts has in these functional diversity. So do the different transcriptomic profiles of fibroblasts identified through single cell work correspond to distinct functions? And then how can we integrate the heterogeneity and the plasticity of the fibroblasts? Are there specific subpopulations of fibroblasts which are activated following infarction and following injury versus other populations that may remain more stable? And which signals mediate these phenotypic transitions? Of course, these are important questions, but however, the ultimate goal is to provide new treatment options for our patients. And a major question in the field is whether and how we should target cardiac fibrosis. And this is a very nice perspective written by two of the experts in the field, Rudolf de Boer and Javier Diaz, suggesting that management of cardiac fibrosis is the largest and medical need in heart failure. And this is true. However, one should keep in mind that fibrosis is quite often reparative and is the outcome of having a non-regenerative organ. So meeting the need of cardiac fibrosis in general may require to develop strategies that actually regenerate the heart. Until then, the question of whether to target cardiac fibrosis and how to target it is more challenging because there is no defined primary disease entity that is cardiac fibrosis. In contrast to the lung or to scleroderma, patients have cardiac fibrosis which accompanies their primary disease, and it may, in many of these patients, reflect a reparative process rather than a source of progressive heart failure. So although in heart failure patients, the extent of cardiac fibrosis has adverse prognostic implications, this could reflect the severity of the primary injury rather than the primary detrimental effects of fibrotic remodeling. So the key questions in the clinic which require clinical research to be addressed is that what is the contribution of fibrosis or fibrotic remodeling to systolic and diastolic dysfunction and to arrhythmogenesis in heart failure patients? And can we perhaps identify patient subsets with maladaptive fibrosis? So certain patients may exhibit dysregulated or excessive reparative fibrotic responses, much like these fibroblast-specific SMAD7 knockout mice that may adversely affect outcome. These patients may benefit from the treatment with antifibrotic agents, in contrast to others who may not benefit. And this is something that we need in cardiovascular clinical care that I think eventually it will develop, is the need to develop pathophysiologically driven patient classifications in myocardial disease. So we have the syndrome of heart failure, which is pathophysiologically very heterogeneous. We have some attempts to classify and stratify patients based on superficial indicators like ejection fraction. So we have HFrEF, HFpEF. You could add mid-range EF and as many categories as you want in there, but it will remain problematic because ejection fraction provides minimal pathophysiologic information. What we would like to have is to be able to classify patients on the basis of the underlying pathophysiologic alteration. And this, in the context of fibrosis, could be achieved with a combined effort that would include clinically defined groups. So, in some patients, for example, diabetic patients, they may tend to have increased fibrosis, perhaps circulating biomarkers, which have their own problems. So there are collagen biomarkers and matrix protein degradation biomarkers that unfortunately do not reflect just the myocardium, but many other organs. And perhaps the more promising of those, but also the most expensive, imaging, and the CMR in particular, through which one could identify specific patients who may have overactive fibrosis and may benefit from targeting fibrosis. And this illustrates how difficult it is to translate concepts into the clinic because this pathophysiologic heterogeneity in patients is a major obstacle. It's important to have dreams and high ambitions. However, in addition to these quixotic dreams, it's important to have a sense of reality and the down-to-earth logic of Sancho Panza. And with these thoughts, I will close the presentation. I would like to thank our funding sources and the members of my lab who did all this work. What I showed today was mostly done by Claudio Humeres, by Isabella Tuleta, and by some past members, Linda Alex and Ruoshvili. And thank you very much for your attention. Be happy to take any questions.